Hi, my name is Angel,
Hi, I'm Jessica.
and today we'll be covering regulation of gene expression in bacteria.
A majority of gene regulation is carried out by proteins, known as regulators, that can bind to promoter sequences. Suppose the bacteria is E. coli. Many of the genes are turned on automatically, known as being expressed constitutively. There are a few main regulators.
One of them is the operon, which controls the expression of a new substrate, and it is able to induce the formation of new enzymes. For instance, transferring E. coli from a glucose medium to a lactose medium may cause it to decline in metabolic activities, then increase. The cells can then produce enzymes that result in the metabolism of the lactose, thus the culture will rapidly grow. An amino acid like tryptophan can also repress enzyme synthesis.
A lactoperon is an example. It's responsible for the metabolization of lactose in E. coli. It consists of three structural genes: LAC-Z, LAC-Y, and LAC-A. LAC-Z codes for B-galactosidase, which cleaves lactose into galactose and glucose. LAC-Y codes the transmembrane protein responsible for the lactose uptake. LAC-A codes for transacetylase, which transfers an acetyl group from coenzyme A.
Tryptophan synthesis requires 5 enzymes, and they cluster together to shut down the operon. These 5 enzymes are co-repressors, and they are responsible for turning off unneeded protein synthesis, and it shuts down operons by binding to the operator.
CAP is another example of a regulator. The recognition helix is responsible for binding to a particular sequence of bases in DNA. Specifically in bacteria, helix-turn helix proteins are used for recognition and binding to a particular sequence of DNA.
Ribose switches are 5' untranslated regions in a molecule of messenger RNA, which has a specific binding site for the metabolites. Some control translation, while others control transcription. That's it.
Thank you!